Welcome back. Today we're going to learn how to do a dihybrid Punnett square or a dihybrid cross. The normal Punnett squares that we did are called monohybrid crosses because mono means one and di means two. So we're going to be crossing two traits instead of one. So there are several steps and here they are right here. The first step is to determine the parent genotypes. So the annotation for genotype are letters. With a dihybrid cross, each parent's genotype is going to be 4 alleles, not 2, like in a regular Punnett square. So you would have like this, not this. So you have big A, big B, little b, instead of just 2 letters. Okay. So the next thing you're going to do is determine the possible gametes donated by each parent. And the way you do that is with the FOIL method, like from math class. So if I had a, big B, little b, I would draw a line down the middle and then do FOIL. FOIL is first, outer, inner, last. So if you look, which are the first letters in their group? It's this one and this one. So I'm going to cross it with first. The next one are the letters that are outer. And so I'm going to use this one and this one. Outer. And then the letters that are inner is little a and big B. And the letters that are last in their group are little a and little b. And you would write those down. So first, again, is big A and big B. Outer is big A and little b. Inner is little a and little b. And last is little a and little b. Now I know one that's confusing is this one because you're like, why is this little letter in front of the big letter? These 2 letters represent the gametes from the parents, and they always go in order. So if A's were first here, then A also has to be first here, and the size doesn't matter. Okay? So the next thing you're going to do is put the gametes on the axis of the chart. So if you look here, this is the new Punnett square. And so gametes would go here and here. And we'll go over that in a moment. And then you would fill in the Punnett square and then cross the possible, not cross, you would determine the offspring phenotypes. We'll do all that in a minute. So step 1, determine the parent genotypes or letters. So it says tall green plant that is heterozygous for both traits is crossed with a short yellow pea plant that is homozygous recessive for both traits. So some information is missing. We're going to fill it in right now. Big T is tall and little t is short. Green and yellow. Okay, so now we can start. A tall green plant that is heterozygous for both traits. So it's heterozygous for tall and it's heterozygous for green. Heterozygous is what? Different. Which means we'll have different alleles for tall and different alleles for green. Which means big T, big G, little g. That's what you should get for parent 1. And that parent is crossed with a short yellow plant that is homozygous recessive for both traits. So homo is same and recessive is small for both of them. So short is little t, and yellow is little g. Okay, so we've done step 1. Step 2 is to determine the possible gametes donated by each parent. And for that, you need to do FOIL from earlier for each parent. So first would be big T and big G. Outer is big T, little g. Inner, little t, big G. And last, little t, little g. And you would also have to do FOIL for the next parent. So you draw your line, first, outer, inner, last. You'll notice that all of these happen to be the same. It won't always be that way. It just is for this problem. And then you're going to do the next step, which is to put the gametes on the axis of the chart. So you just write them down in order. So big T, big G, big T, little g, little t, big G, little t, little g. And I got these from right here with the first parent, these gametes. And then the second parent are these gametes. So I'm just going to write them on the side. Little t, little g for all of them. Okay. Now the last part, or not last part, second to last part, you're going to fill in the Punnett square. So the t's were in the front here. Then they also have to be in the front inside of this box. So you just cross them. So I like to cross each letter instead of the whole thing so that I don't get confused. So which t's should go in this box? Should be big T and little t from here. The next letters that go in the box are the g's. So it's big G and little g, like that. So the next one is here, big T, little t. And now the g's, little g, little g. So the next one over is little t, big G, little g. Again, don't get confused that there are little letters here. The t's always go first because they were first up here. And then the last one, little t, little g, little g. Okay? So you can fill out the rest of the Punnett square, but all of these are the same right here. And so that means vertically they're all going to be the same. So big T, big G, little g. And then these would be the same. And these would be the same. And these would be the same. Okay. So then all we have to do is count. How many are tall and green? So you know anything that's tall is going to have a big T in the box. And anything that's green is going to have a big G in the box. So if we look here, all of these are tall and all of them are green. These are also tall, but they're not green, so it doesn't count. So there are 1, 2, 3, 4 tall green plants. The next one, we're looking for tall, so it's a big T, and yellow, which would be 2 little g's. So these are all tall, and they're also yellow. So that's 4. The next is short and green. If it's short, you need 2 little T's, and green, there's going to be a big G somewhere. Which are these? 1, 2, 3, 4 that are short but green. And then lastly is short and yellow. So short is 2 little T's, and yellow is 2 little G's, which are here. So there's also 4. When you're finished, the number should equal 16 because there are 16 boxes, and that way you know you've answered all of them. You're going to turn this paper over now and practice.